Morning students, myself Sarita, biology lecturer, Government Girls Senior Secondary School, JANDIALA. Today we will study sexual reproduction in flowering plants. So first of all, we should know the various parts of a flower. Flower consists of androecium, that is the male part of the flower, gynoecium, which is the female part of the flower, and both these parts which are present in the flower, they are the reproductive parts, whereas the green part of the flower, which protects it in the early stages in the formation of bud, is known as sepals, and the colored part of the flower, that is known as petals, and these two are non-reproductive parts of a flower. Now we will study what is a flower. Flower is a modified shoot. It develops from the floral primordia. Primordia means outgrowth. So primordia first develops into the floral bud and then it grows into the flower. These are the primordia. So flower is the reproductive part of the plant. It consists of androecium as well as gynoecium. Now we will study the structure of the stamen. So stamen is the male reproductive unit. It consists of filament and anther. Filament is attached to the thalamus or to the petals. This is the internal structure of the anther, which we will study in details. So anther is a bilobed structure. It consists of two lobes and each lobe with two theca. That's why it is known as dithecous. And each theca has microsporangium. So this is anther and it consists of theca and each theca has microsporangium. In cross section, anther is a tetragonal structure consisting of four microsporangia, two in each lobe. Later, two microsporangia of each lobe, they fuse together to form the pollen sac. Now the structure of the anther. Anther consists of microsporangium, which is circular and it is surrounded by four layers. So this is the structure of the microsporangium, and under the microscope we will see these layers. First outer one is epidermis. It is the outermost covering of the anther. Then endothecium. Then 1 to 3 layers of middle layers. Then there is a tapetum. Then there is these cells. These are known as sporogenous tissue. So the function of the epidermis is to protect the pollen and help in dehiscence of the anther to release the pollen grains. The internal layer above the sporogenous tissue is the tapetum. It provides nourishment to the growing pollen grains. When the anther is mature, a group of compactly arranged homogeneous cells, sporogenous tissue, occupies the center of each microsporangium. This is the structure of the pollen grain formed inside the pollen sacs. So now we will study the process of microsporogenesis. The process of formation of microspores from the pollen mother cells or the sporogenous tissue through meiosis is known as microsporogenesis. The cells of the sporogenous tissue undergo meiosis. So these cells, they will undergo meiosis to form microspore tetrad arranged in cluster of 4 cells. Each of these sporogenous tissue has the potential to form a tetrad. So each cell can form a tetrad. So each cell is a microspore mother cell. On maturation and dehydration of the anther, when it dries, the spores dissociate. They will all dissociate and develop into the pollen grains. Pollen grains release with the dehiscence of the anther from the stomium. These openings of the anther, is known as stomium, and the pollen grains will be released. Now the structure of the pollen grain. Pollen grain is a spherical in shape, 2 layers, outer hard one, that is the exine, and inner thin, that is the intine. So exine is made up of sporopollenin, which is resistant to organic matter. It can withstand high temperature, acids and alkalis and enzymes, and it has small prominent apertures which are known as germ pores, where there is no sporopollenin. The internal layer is intine. It is thin, continuous layer and made up of cellulose and pectin. Pollen grain is surrounded by the plasma membrane. Then there are two cells inside the pollen grain. The one is vegetative cell and the other is smaller one is the generative cell. Vegetative cell is the larger one. Abundant food reserve is there and a regular nucleus is there. Generative cell is small, spindle shaped, and it floats freely in the vegetative cell of the cell cytoplasm. Mostly in case of 60% of the species, pollen grains are shed in 2-cell stage, wherein in 40% of the species it is shed in 3-cell stage, in which the generative cells divide mitotically to form the 2 male gametes. So the total number of the cells will be 3. Now all these stages we have studied, they are the pre-pollination events. And now we will study the female reproductive unit. So the female reproductive unit is female is the reproductive part of the flower. Pistil is the reproductive part of the flower. It may be mono, bi, tri or polycarpillary, means that it may be 1, 2 or 3 or more than 3. It may be syncarpous. There are 2 or more carpels are fused together to form the compound ovary, for example in petunia. It may be apocarpous. Apocarpous means 2 or more carpels in free state, and for example in buttercup. Each pistil consists of ovary, style and stigma, whereas the stigma is the landing platform for the pollen grains, and the style raises the pistil to increase the chances of pollination. Ovary has 1 or more cavities called the locules. If we see the cross section, we will see the placenta and there are locules, single locus. Placenta in a locules were ovules. Number of ovules may be 1 or more. Now, how ovule is formed? Ovule arises from the primordia on the placenta as an outgrowth. There is a short stalk which attaches the ovule with the placenta, that is known as funicle. Primordial grows into a mass of cells forming the nucellus. That is the body of the ovule. There are two protective coverings around the nucellus. These are known as integuments. So these are the integuments. There is a small opening which is not covered by the integument, that is known as micropyle. So these are the integuments covering the nucellus, and this is the micropyle. The lower portion or the basal portion of the ovule is known as classa. It lies opposite to the micropyle. So this is the micropyle, this is the classal end. There is a single embryo sac or the female gametophyte located in the nucellus, which is developed from the megaspore. Now we will study how this embryo sac is formed inside the ovule. So one of the nucellar cells in the micropylar region is differentiated into megaspore mother cell. This cell is large, contains dense cytoplasm, and prominent nucleus is seen. It undergoes meiosis, reductional division, to form 4 haploid cells, megaspore tetrads. 3 megaspores will degenerate and only one will form the megaspore, which is the functional megaspore. So this functional megaspore is the first cell of the female gametophyte. Its nucleus undergo mitosis to form 2 nuclei, then which will move in the opposite directions to form 2 nucleate embryo sac. Then it will undergo 2 successive divisions or 2 more mitotic divisions to form 8 nuclei. So this is the structure. So this is the embryo sac with 1 nucleus, then 2, then it will again divide to form 4, then it will form 8. 3 will remain towards the micropylar end, 3 will remain towards the classal end, and 1 nuclei from both the poles will move in the center and form the polar nuclei. This is the development of the female gametophyte. Now male gametophyte is formed, female gametophyte is formed. The male gametophyte has to reach the female gametophyte for the fertilization, and the process by which it will reach, that is known as pollination. So next steps are the pollinations. So first of all, we should know what is pollination. Pollination means transfer of the pollen grains from the anther to the stigma is known as pollination. It is of 2 types, self-pollination and cross-pollination. Autogamy, self-pollination, and allogamy or xenogamy, that is the cross-pollination. So what are the devices that favor autogamy? The flowers, there is a simultaneously maturation of the anther and the pistil. Anther and the stigma, they lie close to each other. Mostly in case of the closed flowers, they are like clistogamous flowers like groundnut, oxalis and viola. These are clistogamous flowers. So in this case, the flowers, they never open to expose their sex organs, and the pollens fall on the stigma of the same flower. So in this case, there are no chance of cross pollination and no pollinators are required in such cases. Now cross pollination. So what are the devices that favor the cross pollination? The flowers are mostly unisexual, dioecious, and pollen grains and the stigma receptivity are not synchronized. There is different orientation of the anther and stigma of the flower. That means the two or more types of flowers having different length of styles and stamens. In case of jasmine, primrose, and there is self-incompatibility. This means that the pollen grains of a flower are incapable of affecting fertilization even if they are placed on the stigma of the same flower due to mutual inhibition. The these pollen grains of one flower, they don't grow on the stigma of the other flower because there is mutual inhibition between them. Now there are various agencies which will help in cross pollination. First of all, that is the wind pollinated flowers. Wind pollinated flowers, or the wind will help in pollination. So the process, this type of pollination is known as anemophily. Pollen grains are light, they are non-sticky, dry, and they are winged. They have well exposed anthers. Large feathery stigma is present here. So this is the stigma. Flowers arranged as inflorescence in groups, and there is a single ovule. Now next, water pollinated flowers, that is known as hydrophily. So mostly seen in submerged flowers like Valsinaria hydrilla. In this case of Valsinaria, male flowers are released and on the water surface, and the female flowers reaches the surface for pollination, and mostly the pollen grains are covered by the mucilage. Now in case of insect pollinated flowers, it is known as entomophily, pollination by insects. Flowers are large, they are mostly beautifully colored or brightly colored and showy. If flowers are small, they are grouped in inflorescence, highly fragrant. They have nectar, and mostly they have sticky pollen and stigmatic surface. And moreover, they provide the insects with nectar, food, and they provide safe place for laying eggs. Now the next is most important part of this lesson, that is pollen pistil interaction. When the pollen grain reaches the stigma, that interaction is known as pollen pistil interaction. There is recognition of compatible pollens. It is interaction between the chemical components of the pollen and that of the stigma. Now there are the steps, that is the germination of pollen and the development of the male gametophyte. So compatible pollen starts its germination stimulated by certain secretions of the stigma. So pollen grains, they land on the stigma, and this stigma secretes chemicals to attract the pollen grains. So intine grows out through one of the germ pores. The internal covering, that is the intine, it grows out through one of the germ pores. Now contents of the pollens moves into the tube. Vegetative cell as well as the generative cell, or if it has divided, it will form two male gametes. Then two male gametes, they are moving in the tube or the pollen tube, which is formed by the intine. Now next, pollen tube grows through the tissue of the stigma style by secreting enzymes to digest them and enter the micropyle of the ovule. It enters the embryo sac through the filiform apparatus and enters one synergids to liberate the male gametes. Germinated pollen grains with pollen tube carrying vegetative nucleus and two male gametes is the fully developed male gametophyte. Now the next important step is the double fertilization. There is release of two male gametes. Pollen tube, when it enters the micropylar end, it releases the two male gametes from pollen tube into the cytoplasm of the synergids. Fusion of one male gamete with the egg is called syngamy, and it forms the zygote, which becomes diploid, and it will form the embryo. Fusion of the second nuclei will be with the polar nuclei of the center cell to form 3N structure, that is known as PEN, primary endosperm nucleus. As syngamy, fusion of the male gamete as well as the pollen male gamete will form one type of fertilization, and second male gamete that formed a triple fusion, it occurs in the embryo sac, therefore it is known as double fertilization. Two fertilizations occurs in the embryo sac. Central cell with the primary endosperm nucleus is called primary endosperm cell, which develops into the endosperm. So this will form the endosperm, this will form the embryo. This is triploid 3N and embryo is 2N. So this is the complete process of double fertilization. Pollen grains landing on the stigma, it will form the pollen tube. It contains the sperm cells, male gametes. It will enter the micropylar end. One will fuse with the egg nucleus, the other one will fuse with the pollen nuclei. So the double fertilization occurs in angiospermic plants. Now, after the double fertilization, there are post-fertilization events. That is the development of endosperm from the polar nuclei. Then there is the development of the embryo from the zygote. Then maturation of the ovule into the seed, maturation of the ovary into the fruit. These are the post-fertilization events. Now, the development of the endosperm. The formation of endosperm is initiated by mitotic division of the primary endosperm nucleus, which is 3N. There are three methods of embryo development. One is nuclear, cellular, and halobial. So in case of the nuclear type of endosperm, the first division of the primary endosperm nucleus and there is nuclear divisions are not accompanied by the cell wall formation. Nuclei are produced and free in the cytoplasm. This is a nuclear type of endosperm formation. In the second is the cellular. In case of the cellular, there is cytokinesis. After each nuclear division, there is a formation of cell wall. Third type is halobial. So in this case, it is an intermediate between the nuclear and the cellular. There is a formation of nuclei as well as there is a formation of cell wall around the nuclei. So both the divisions occur in halobial type of endosperm formation. So what is the main function of the endosperm? It provides food to the developing embryo. If endosperm is completely utilized in seeds, then such seeds are non-albuminous. So if there is endosperm is present, then it is known as albuminous. So for example, in wheat and rice, seeds are endospermic with endosperm stored. So non-albuminous seeds like pea and gram, the food is stored in the cotyledons. So these are the diagrams of the nuclear endosperm development. This is the cellular and this is the lobial. So there is a development of the embryo. So the ovule, it develops and forms the mature seed, and the embryo is present inside the seed. So we will study the development of the embryo. After the zygote is formed, it will start formation of the embryo. Zygote will divide mitotically to form the proembryo. Then it divides into globular form, heart-shaped embryo, horseshoe-shaped embryo with one or two cotyledons. If one cotyledon is present, it is called monocotyledonous seed development, embryo development. If two cotyledons are present, then it is dicot embryo development. Now the last portion of this topic, that is the life cycle of the angiosperm. It is a summary of the entire lesson. So here we will start from the flower. So and this is the male part of the flower. These are the stamens with anthers, and this is the filament. In the anther there are microspore mother cells. It will divide by meiosis to form 4 microspores, and these microspores are the pollen grains. It has two cells. One is the vegetative cell and the other is the generative cell. Then there will be formation of pollen tube and tube nucleus, as well as the generative nucleus will pass in the pollen tube. The generative nucleus will form the 2 male gametes, and this is the male gametophyte. Now on the other side, the female part, it consists of ovary, style and stigma. In the ovary there are ovules, and in ovule there will be a nucellus. There, by the process of meiosis, it will form embryo sac, and 3 will degenerate and 1 will form the embryo sac, and the nucleus will divide by mitosis. It will form 8 nuclei. 3 will remain towards one pole, that is towards the causal end. 3 will move towards the will remain in the micropylar region. 1 from each will come in the center and form the polar nuclei. So by process of pollination, the male gametophyte will reach the stigma of the ovary. It will pass from the stigma to the style. Pollen tube will pass and reach the ovule through the micropylar end by entry through the micropylar end in the ovule. Here the male nuclei, one will fuse with the egg nucleus, and the other one will fuse with the polar nuclei. Polar nuclei will become triploid, and the egg nucleus and the male nucleus, they will fuse to form diploid structure, that will be the zygote. Triploid structure, PEN, primary endosperm nucleus, will form the primary endosperm cell, which will form the endosperm and provide nourishment to the growing embryo. Now embryo will develop inside the ovule, and after its development, embryo will form the new plant. Endosperm will provide food to the embryo, and the embryo will form the new plant. Again it will bear flowers, and the same cycle will repeat once again. So this completes sexual reproduction in flowering plants. Thank you.